Hello, everyone. This is Jiang. Thank you so much for joining my talk. As a brief introduction, I'm a co-founder and director of scientific affairs within Vizgen. So first, I wanted to really thank the organizers for putting together this meeting. And I'm really excited to have the opportunity to share with you what we're doing here at Vizgen. So for today's talk, I hope to first give you a sense of why spatial genomics is such a powerful technique. And then I'm going to walk you through the technical aspects of our platform technology and how it differs from some other spatial technologies. And finally, I will also use a couple of showcase examples to illustrate how you can apply these technologies to address different biological questions. As some of you may know, spatial genomics is actually quickly becoming an essential tool for biological research. And we're truly excited to see what new biological insights and discoveries it will enable and the impact it will bring in the near future. So just to give everyone some context first, we know that biological tissue are quite complex and are made of many different cell types that express genes very differently. Understanding that gene expression profile within each cell and across the tissue is quite critical for better understanding of the entire biological system. As you may know, over the past 2 or 3 decades, we have seen a revolution in genomics research, which really enables us to characterize the biological system at unprecedented scale and resolution. As you can see from this slide, the landscape of genomic evolution from bulk RNA sequencing to single-cell sequencing, we can now start to interrogate the gene expression profile of individual cells and really use this information to understand the cell composition as well as cell-cell interaction. However, sequencing-based technologies actually do have a fundamental limitation because they don't really provide any special context of what you're looking at. To do the sequencing, cells either have to be destroyed or dissociated, and thus we lose the rich spatial information for the intricately organized 3D biological tissue. And as you now can appreciate, many biological problems do require multiplexed, spatially resolved, and single-cell measurement. We really see a huge demand for technologies that can satisfy this criteria, and that's what Vizgen is really working on. We aim to bring the best spatially resolved single-cell genomics tool to the research community and for clinical research in the future as well. So our platform technology licensed from Harvard University is called MERFISH, which stands for Multiplexed Area Robust Fluorescence in situ hybridization. This is an RNA imaging-based technique that can locate and quantify different RNA species at the same time with subcellular resolution. As some of you may know, that there are actually existing RNA imaging technologies such as single-molecule FISH. However, the limitation of these traditional technologies is that the gene throughput is relatively low. Oftentimes, you can not profile more than 5 gene species at the same time. Multiplexing is very challenging for these traditional methods because it relies on the use of spectrally distinct fluorophores to report different RNA species. And since we don't have hundreds of thousands of different dyes available, then it's very difficult to do hundreds of thousands color imaging. While in contrast, MERFISH really enables highly multiplexed RNA imaging capable of resolving hundreds to tens of thousands different RNA species within cells. So as you can see here, there are 3 key aspects that really make this highly multiplexed measurement possible, which is complementary labeling, sequential imaging, and area-robust barcoding. To perform MERFISH, as you see on this slide, we use a complementary labeling strategy to label different RNA species with a type of oligoprobes that contains different barcodes. Then each barcode is going to be detected through fluorescence imaging by sequential round of imaging. And the fluorescence on and off pattern generates an optical barcode, and we use this to help resolve different RNA species. This spatially resolved RNA profiling data really gives the physical picture for the cell or tissue of interest, as shown by the image on the right, with now colored dots indicating different RNA species and gray line indicating different cell boundary. So MERFISH was initially invented by our scientific co-founder, Dr. Xiaowei Zhuang at Harvard University in 2015. And since the initial publication in the Journal of Science, it has been validated by a long list of publications, either from Xiaowei's lab or from some other labs, and has already brought many transformative discoveries. And actually, spatially resolved transcriptomics was selected as method of the year in 2020 by Nature Methods, and MERFISH was featured there. It really highlights the excitement it has already generated so far. So furthermore, around the world, there are already many institutions who are already adopting MERFISH either independently or through participation of Vizgen's Early Access Program. This includes 10 more institutions, not even listed on this slide. So within Vizgen, we are actually making MERFISH more broadly available through our commercial platform solution called MERSCOPE. In early March, we announced the commercial launch of our MERSCOPE platform. This platform facilitates the whole MERFISH measurement with a complete reagent, instrument, and software solution. It combines the advantages of MERFISH technology with a streamlined workflow as shown here, from the chemistry reagent side, gene panel design, to downstream instrument, hardware, and software. And using this, we maintain the sensitivity, higher multiplexing capability, as well as high resolution as established by MERFISH. And this really makes the whole system easy to use. So here is a summary of MERFISH technology's key features. Looking on the right, first, MERFISH has high multiplexing power. Through academic publications, MERFISH actually has been demonstrated up to 10,000 genes. But in our first generation of reagent offering, we measure hundreds to thousands of genes simultaneously in single sample. Secondly, MERFISH has exceptional spatial resolution. We can localize RNA transcripts with subcellular resolution. This really allows us to zoom in into individual cells and directly analyze the spatial distribution of the entire transcriptome. Or you can even zoom out and start to analyze the molecular and cellular profile of the entire tissue at biologically meaningful length scales as well. Thirdly, MERFISH has high sensitivity. Since MERFISH is based on single molecule FISH, which is often considered as the gold standard for quantifying RNA copy numbers in cells, so MERFISH also maintains that single molecule sensitivity. And for people who work in the single cell field, they can probably start to appreciate why sensitivity and detection efficiency matters, because many lowly expressed genes don't often have too many counts in this measurement. And MERFISH's high detection efficiency really enables us to detect those lowly expressed but functionally important genes very easily. And furthermore, MERFISH has very high cell throughput. We can profile square centimeter sized tissue with hundreds of thousands of cells in a single experiment. No follow-up sequencing is needed. You get all the information in a single experiment run with no additional sequencing cost. And finally, MERFISH is very flexible, and we can perform MERFISH on almost any tissue or sample types. So as a gene expression profiling tool, as you can imagine, MERFISH at its basic form reports the location and quantities of RNA species, as well as the cell boundary information. But with so much information, it really opens up many application areas. For example, we can use this tool to profile the gene expression profile in situ, map and catalog cell types and state in tissue, and map the intracellular organization of the transcriptome. Furthermore, you can even use this tool to do imaging-based genetic screens such as CRISPR screens, or apply it to image DNA and trace chromatin structure. So in the next few slides, I'm going to walk you through a couple of showcase examples to really illustrate how we can use MERFISH to gather biological insights. So the first showcase illustrates how MERFISH could be used to profile gene expression profile in situ across very large tissues. As we know, to understand tissue function, it is critical to understand how different genes are expressed at different anatomic sites and how the gene expression profile change during disease onset, progression, and after drug treatment. So in this showcase example, we used MERFISH to profile around 500 genes to map out the major cell types and cell receptors in the mouse brain. The gene panel, as illustrated here, include around 50 canonical marker genes and around 433 cell receptors, including GPCRs and RTKs. On the slide, what you are seeing in the left in this 9 by 7 millimeter image, what you are seeing is the distribution pattern for a subset of 8 genes out of this 483 gene panel. Hopefully, by glancing at the picture, I hope you can already see the very intricately layered structure in the mouse brain. For example, across this corner section, you can even zoom in to each single field of view as shown in the image in the middle now to get a high-resolution view of the gene expression profile for individual cells. So all 483 genes are plotted in this image. You can really start to interrogate the gene expression within individual cells. As shown by the plot on the right, this measurement is highly quantitative and reproducible. For example, by looking at the correlation plot with bulk RNA-seq data here, we got correlation coefficient of around 0.88. And in between biological replicates, we got a correlation coefficient of 0.99. And what's really important to note is that MERFISH can very accurately profile genes with a very dynamic range of expression profile, including genes expressed at less than 1 copy per cell. As you are seeing in the correlation plot with bulk RNA sequencing there, the expression range for those profiled genes spans 4 orders of magnitude. And many genes with FPKM below 0.1 or even 0.01 can be very accurately measured. So this high detection efficiency of MERFISH really started to allow us to characterize lowly expressed but functionally important genes. Just to highlight how this actually manifests in real data, we can look at a quick comparison between MERFISH measurement and a measurement coming from an oligoarray capture-based technology. So here, we're considering matched brain samples between these two technologies. As you're seeing by the images on the left, half of the brain slice are shown over there, and we're comparing the counts of transcripts per gene. In the middle, for the same gene panel measured, hopefully you can see the very striking comparison. We found that actually MERFISH consistently detects 70-fold more copies of each transcript. To really put this into context, MERFISH has been demonstrated to have a detection efficiency of around 95% in cultured cells. So for a gene that expressed 100 copies of transcripts per cell, MERFISH can get around 95 copies of that transcript of that particular gene, while the array-based platform can only detect 1.5 copies per cell. So you may start to wonder, what does these numbers mean in real biology, and how does that relate to sort of addressing certain biological questions? Let's really start to zoom in more into this data set and look at one moderately expressed gene, opium receptor delta 1, OPRD1. So in the middle, again, in the mouse brain, with MERFISH's high detection efficiency, we can clearly see the spatial organization of this gene in different brain regions. However, if you look at the distribution pattern from the oligoarray-based technology, there are only a few grid spots with 1 or 2 counts, and then there is no clear spatial pattern. So imagine if you actually studying the drug effects on opium receptors, how much information will actually be missed by using this oligoarray-based technology. Furthermore, if we look at the right, MERFISH measurements actually offers true single-cell resolution, while in the array-based platform, each grid spot is approximately the size of 10 cells with a grid spot size of 55 microns. So by looking at the zoomed-in image of a single field of view from MERFISH measurement, you can clearly see where the OPRD1 gene is expressed in individual cells, while that information is completely missed in array-based platform. So this single-cell resolution of our platform technology really allows us to perform single-cell analysis and atlas the cellular composition of complex tissues. So again, in this same measurement, by counting the number of transcripts with each segmented cells and then clustering the cells based on the gene expression, we can perform the same sort of analysis that you can do with single-cell sequencing. For example, shown on the left, we can identify cell types and project the different cell clusters identified on this UMAP plot. But since this is a spatial measurement, we know that each location is recorded, and then the exact position of each of these cells is also illustrated on the images on the right. And hopefully you can really start to see how we can use and leverage MERFISH for creating cellular atlas. So if you're interested in learning more about this data set, this is the data set we are releasing publicly. It is the measurement of 9 full chronal slices of the mouse brain with this 483 gene panel I just described, which profiles all the clinical cell markers and cell receptors like GPCRs and RTKs. And this could be very relevant for many people who are studying and doing drug development research or neurological research. So to get notified of when this dataset is available, you can sign up on our website. So next, the second showcase example I'm going to walk you through to illustrate how we can actually characterize cell function and state in complex tissues. This is actually a study published in Science back in 2018. So here, MERFISH was used to profile a panel of 155 genes for a total of 500,000 cells in the brain region called mice hypothalamus. So in this study, the gene expression profile information among each cells were used for clustering as I just described. So I will be brief here. And again, as shown by the T-SNP plot here, around 75 cell clusters were identified in this brain region. And you can see different cell clusters for excitatory and inhibitory neurons. On the right, you can see the highly differentiatedly expressed genes for unique cell clusters. On the left, the correlation plot, MERFISH agrees very well with single-cell RNA-seq. Actually, as you can see from this correlation plot here, all the major cell types found by single-cell RNA-seq were recapitulated by MERFISH measurements. Actually, in some instances, as shown by the correlation plot on the right, MERFISH was even able to resolve different cell clusters that were not distinguishable by single-cell RNA-seq. For example, there are two clusters on these correlation plots that are grouped as a single cluster. However, MERFISH was able to segregate that into two different clusters. And this was really made possible by two key features of our technology. First is the high cell throughput. By profiling more samples, more cells, rare cell types will be more easily captured. And secondly, this is really enabled by the high detection efficiency, which allows not expressed but functionally important genes to be profiled. To really further illustrate the high detection efficiency of MERFISH in this study, the authors actually compared the RNA copy numbers detected by MERFISH versus single-cell RNA-seq technology using Chromium. So as shown by this bar chart here, MERFISH consistently detects 7 times more transcripts than single-cell RNA-seq. And this really allowed the authors to profile many not expressed genes that can specify cell type, including neuropeptides. And finally, again, as the cell position is recorded in this measurement, MERFISH could further be used to catalog and reveal the cell organization in tissues. Shown here on this slide now is the cross-section images of the mouse hypothalamus, with each circle now indicating the position of all the major cell classes. From the anterior to posterior of this brain region, I hope you can appreciate the fact that the distribution of different cell types are very heterogeneous. Furthermore, since MERFISH could be readily used to profile different cell types and state, by measuring the gene expression of an early immediate gene called C-FOS, in this study, MERFISH was able to identify which neuronal types are specifically activated during individual behaviors, such as mating, parenting, and aggression. As you can see, aggression behaviors, as you can see in the bar chart on the top, that there are certain neuronal clusters that are expressing C-FOS much higher than the others upon initiating those social behaviors. And of course, since this is a spatial measurement, you can actually map out where these cell types are in the brain, as shown by the images. At the bottom, you will see where those cells are responding to different social behaviors. In a short summary, I hope this example really demonstrates the power of MERFISH for cell atlasing. And this application actually has profound implications for biomarker development, mechanism of action studies, to name just a few here. And it will really provide a lot of valuable insights in understanding complex tissue types and cell-cell interactions as well. So the third showcase example that I am going to walk through illustrate how to use MERFISH to map a more clinically relevant sample, the tumor microenvironment. And the example that I'm showing here is human colon cancer. Here we used MERFISH to profile 417 genes in colon cancer. And what you're seeing on this slide on the left is the spatial distribution of 10 RNA subsets out of this 417 gene panel measurement. With just a few genes, I hope you can already see the rich spatial information for different genes within this particular tumor slice. For example, in the zoomed in images here, we see a group of SOX9 positive tumor cells toward the top, and this group of SOX9 expressing cells encircles SPP1 expressing stroma cells. And at the bottom, you're seeing that adjacent to these tumor cells, there are a large population of B cells marked by the IgHA1 gene. And on the right, since we do have single cell resolution, you can keep zooming in and try to analyze and interrogate the gene expression within individual cells as well. These are some of the things that we started to see with just a few genes, but since this measurement contains 417 genes with single cell resolution across the entire tissue, so we can again perform cell clustering analysis as I just described a moment ago and identify different cell types within the tissue. For example, here this is showing all the major immune cells, including B cells, T cells, NK cells, dendritic cells. And again, the spatial distribution of these different immune cells are illustrated on this slide. For example, you're seeing CD8 T cells, CD4 T cells more enriched at the bottom of this tumor slice, and then a subset of CD4 cells, Th17 CD4 cells, are more enriched at the top. So the final example that I wanted to share with you shows how you can use MERFISH to directly analyze the intracellular distribution of RNAs. As some of you may know, that this intracellular organization of transcriptome underlies many fundamental biological processes such as cell migration, synapse formation, or even tumor metastasis. A better understanding of this organization will definitely help illuminate how these cells function. So in this recent poster study on bioRxiv, MERFISH was used to profile 4,200 genes in individual neurons. And by using immunostaining to trace neurites, MERFISH enables a direct visualization of different RNA species in different neuronal compartments such as axons, dendrites, and soma. And this really allows the identification of RNAs enriched in different neuronal compartments. For example, in the plot showing on the left, what you are seeing are the RNAs enriched in the dendrites. Furthermore, by characterizing the spatial distribution of different RNAs within the neuronal compartments, the authors were even able to find distinct group of genes that shows a spatial clustering effect as highlighted by the heat plot here. For example, we have two clusters, cluster 1 and 2 genes, seem to colocalize with synapses. And very interestingly, these two groups of genes seem to be dependent on protein synthesis, depends on protein synthesis effect. And to some extent, it is also differentiated dependent on the synaptic activity as well. For example, if you look at the bar chart on the bottom right, you're seeing that when we inhibit the protein synthesis with pyrimycin, you can see a fraction of class 1 and 2 genes no longer colocalize with the synapses. In addition to all the showcase examples that I just illustrated, MERFISH has been used to label genomic loci in individual cells and perform genome-scale chromatin tracing. Or you can even use this to perform imaging-based genetic screens such as CRISPR screens. However, for the sake of time today, I'm not going to go through these examples in detail. If you're interested in learning more, I really strongly recommend you to look for the papers that are cited below. So I hope these few examples have given you a good impression of what MERFISH is capable of, and within Vizgen, we keep pushing the limit of the technology by expanding its application areas. So far, we have proven MERFISH in more than 20 human and mouse tissue types, and a few representative tissue types are shown on this slide. In addition, we aim to make MERFISH compatible with any sample type and tissue type as possible, and we have validated MERFISH in cultured cells, suspended cells, fresh frozen, fixed frozen, and even FFPE tissue blocks. We really hope that this versatile tool could be really added into the toolbox of any researcher and really help accelerate their research. So this slide shows the overall workflow of sample prep, and it's a very straightforward process. Briefly, you can place multiple pieces of tissue or cells onto our cover slip, and then the sample undergoes several steps of treatment, including permeabilization, hybridization, gel embedding, and clearing before putting on to our instrument for automatic imaging. And this protocol is actually very similar to a standard ISH protocol you will see, and with hands-on time of around 1 to 2 hours in total. And we envision that any researcher can actually adopt it very easily. So in short summary, I hope that this presentation gives you a good overview of what we are doing here at Vizgen and what our imaging platform looks like and how you can use this to gather biological insights. We are truly excited about the potential of our technology and would love to hear how we could potentially help with your research as well. So on this slide, what you are seeing is our leadership team, and we always would love to hear feedback from your side. And with that, I wanted to thank you again for setting up the meeting and for joining the meeting. And if you have any questions, please let me know or send inquiries to the email address shown on this slide. Again, thank you very much for listening.